Novel gene-based breeding versus field-based breeding in improving fiber quality traits in cotton. So just a little bit of background about Gossypium. There are two cultivated species of cotton in the United States. Last year, an estimated 20 million bales of cotton were produced. The first type of species is Gossypium barbadense. It's called Pima cotton, also referred to as extra long staple because of its high upper half mean length, which exceeds 35 millimeters, which is about 1.38 inches, and fiber bundled strength. And the second cultivated species is Gossypium hirsutum, also known as medium staple upland because of its upper half mean length, which ranges from 27 to 29 millimeters, which is about 1.06 to 1.14 inches, and its fiber bundled strength that is in the range of 26 to 28 grams per tex. Gossypium barbadense in the United States account for about 5% of total cotton production, and hirsutum, which is upland cotton, accounts for 95% of U.S. cotton production. So the focus of my project is on fiber quality. So why fiber quality? The first reason is that improved fiber quality means improved yarn and an improved fabric, a better fabric. So there's an improved product portfolio potential resulting from improved fiber traits. The second reason is to remain competitive with man-made fibers, especially these days. The competition from Under Armour and Lululemon and all that is really high compared to the cotton fabric. And the third, and probably one of the most important reason why we want to focus on fiber quality, is to maintain the export market. The international markets demand higher fiber quality measurements when compared with U.S. cotton. And on the right side, you'll see the little table that gives you a comparison of the international standards when it comes to cotton fiber quality and the U.S. standards. And the international standards are slightly higher, so we need to maintain our export market because about 65% of U.S. cotton is exported. So as I said earlier, the goal is to improve cotton fiber quality. Now there are a lot of traits that are related to cotton fiber quality, but the two main traits that we're going to focus on are upper half mean length and fiber bundle strength. Upper half mean length means that is the average length of the half of the longest fibers in a cotton sample. And fiber bundle strength refers to the amount of weight that is required to break a 1,000-meter long bundle of fiber. So, as I told you earlier, that my study focused on two comparative methods. The first one is the first selection methodology that we're going to look at is field-based breeding. In other words, the traditional way that the cotton breeders have selected individual plants for superior fiber quality. Its methodology is pretty simple. You go out to the field, you take individual plants, you do a little visual screening, you collect fibers from different position bolls, and you compare them and look at, as you can see it in the picture, you put it on your finger or whatever tool that you want to put it on, and see that if this is a long fiber or compared to the rest of the individual plants, then I'm going to keep the longest fibers, and then take those selections to progeny rows next year. On the individual plant selections that are done in field-based breeding, we're going to collect the high-volume instrument measurements for upper half mean length and fiber bundled strength. HVI is the most accepted standard of measurement system in cotton. Basically, the U.S. cotton farmers, when they sell their cotton, they are using this system that is going to give them any premium or discount when they're selling their cotton in bales. And then from there, we're going to take the top 10% and the bottom 10% fiber quality selections and advance them to progeny rows for comparison purposes. The second methodology is, as Dr. Hongbin proposed, called gene-based breeding. And the definition of gene-based breeding is breeding that is not only based on alleles, also gene allele selection, but also on the expression profiles of the genes controlling the targeted traits. This diagram will help us understand better what the gene-based breeding methodology is. To your left is marker-assisted selection, or genomic-assisted breeding, as we call it, where you can see that there are two markers that are identifying or that are locating where the gene is in the genome. Whereas in gene-based breeding, the marker itself is part of the gene sequence. So in gene-based breeding, we are using these so-called gene-based SNPs, which are related to our fiber quality traits, mainly upper half-mean length and fiber bundle strength. And then we're going to use those gene-based SNPs to screen our individual plant selections in order to determine which SNPs predicted the better or the superior fiber quality lines and which ones didn't. This is just a couple of bullet points on how the methodology, how the gene-based breeding protocol was developed. I was not part of this development process. This was done a couple of years before this project was started. But the main takeaway from this is that about 474 genes were identified for upper half-mean length, and about 1,100 genes were identified for fiber bundle strength. Now, it's important to note that these genes are not all positively linked with upper half-mean length. Most of them have a negative relationship with upper half-mean length and fiber bundle strength. So moving on to materials and methods. So we wanted to look at two different populations, so that we can have two different sort of comparisons, two different type of populations for our individual plant selections. Our first population was an F2 generation population. Now these populations were grown in 2016. So in 2016, the F2 population was grown. It was an interspecific population. That means it had Pima introgression, barbadense introgression in it. And the reason why we wanted to use this F2 population was because we expected high polymorphism, because interspecific background brings a lot of genetic variation, and we thought that using these 11 gene-based SNPs can help us separate our top producing fiber quality lines to our poor quality fiber quality lines. The second population was an F3 generation population that we used in 2016. This is an intraspecific population because it's an upland by upland combination. Both the parents are elite parents or fiber quality. And this type of population is what the cotton breeders are interested in, mainly because almost all of the cotton breeders who want to breed for superior fiber quality are almost never going to touch barbadense when it comes to breeding elite combinations for fiber quality. This is I'll sort of give you on how we did the selections, how it was divided. So for the interspecific population, we made 250 individual plant selections for field-based breeding. That's where we went out to the field, we made our selections, and then we phenotyped. Then we did the HBI measurements on them and got their phenotype and selected the top 10 and the bottom 10. And then we made 250 individual plant selections for gene-based breeding. Now these 250 plants for gene-based were randomly selected for the interspecific population, it was about 2,000 plants. So 250 plants randomly selected from 2,000 plant breeding nursery for interspecific population. And for intraspecific, it was the same number that we selected. The only difference was that in our intraspecific population, we had about 3,000 plants where we randomly selected our 250 plants. And the top 10% and the bottom 10% were then again decided from our SNP, gene-based SNPs that we used to make our predictions. This is just an example of our selection screening, where our gene-based SNPs were used to score individual plant. So on the leftmost column, you'll see that F3DBG203, when it was screened with this gene-based SNPs that we used, it had a particular score. favorable allele got a 2, unfavorable had score of 0, and heterozygous got 1. And that's how we scored our 250 plants. And then we decided the top 10% and the bottom 10% in the gene-based. And in the field-based, it was really easy to make our selections because we actually had our HBI data. We were not really predicting. We actually had the measurement from where we just selected top 10 and bottom 10. So when we made our selections in 2016 for our the random selections among our gene-based, we wanted to see that whether or not did we actually capture any variation. That these were randomly selected, so we wanted to see what was the range of upper half mean length values that we were dealing with. And also in today's presentation, I'll be mainly focusing on upper half mean length only. So in upper half mean length for an interspecific population, you can see that it ranged from 1.1 to 1.64 inches. So that was a good range of upper half mean length values that we were dealing with. So we thought that, okay, I think we have a good enough of a variation, a sample size where we can select the top 10 or the bottom 10. For intraspecific population, it was the distribution was, or the range was slightly narrower, but then enough to have a good amount of variation that we can select from. So out of those 474 genes for upper half mean length and 1100 for fiber bundle strength, we were able to use only 11 gene-based SNPs for fiber bundle strength and upper half mean length to screen our gene-based breeding selections of 250 plants, and from which we determined the correlations of gene-based selections with their actual HBI phenotype. And with our field-based, it was the visual determinations in the field that we made, and from which we determined the correlations with its actual HBI phenotype. The experimental design, so the selections were made in 2016, we advanced them in 2017, and then we did an RCBD design in 2018 and 19. So interspecific population was of F2 generation in 16, and in 2018 and 19, interspecific population was in the F4 generation, or the selections were in the F4 generations. And for the intraspecific population, it was the F5 generation in 18 and 19. And so the first RCBD trial had top 10% and the bottom 10% of both gene-based and field-based planted in 2 replications. From where it was in College Station at the AgriLife Research Farm, normal cotton production practices were followed, hand harvested 30 open bolls and ginned, and then the lint samples were evaluated, and from where HBI fiber properties were determined mainly for upper half mean length and fiber bundle strength. So let's look at the first correlation graph. Now this is our gene-based SNP predictions plotted against its actual measurement, its actual HBI phenotype. So the x-axis is our SNP predictions, upper half mean length in inches, and our y-axis is the actual HBI phenotype. Now if you look at the top 10% over here on the right side for 18 and 19, the R-square values are 0.49 and 0.34. Now these are not, this is an acceptable R-square value considering that these were randomly selected and we were able to get a correlation of 0.49 or 0.34. However, these were supposed to be the top 10% among our 250 plants. So we should have seen more clustering towards this on the top or on the higher end instead of a little bit in the middle. Similarly, with the 2019, it was the same story. We should have seen something on the higher end, but then it was in the middle. So these 11 gene-based SNPs were not conclusive enough to establish a relationship that we can make a prediction in the in the F2 generation, and then and can correlate well with the next year progeny row. Now this is for the interspecific population. For the bottom 10%, once again, acceptable R-square values for random selections. However, there's a lot of top values floating around in the bottom 10%. Similarly with 2019 as well and 2018. So in the interspecific populations for upper half mean length, these 11 genes were not conclusive to make a prediction in order to get a good upper half mean length value. Sorry. So this is the intraspecific population. These are again SNP predictions plotted against their actual HVI phenotype. For the top 10%, even though there are top values in the top 10%, but then looking at the R-square values for both years, and just by visually looking at it, you can see that there's no relationship that we can determine. For the bottom 10%, even though the R-square values in 2018 was 0.52 and in 19.35, actually the bottom 10% had pretty much all the higher fiber quality lines. And we can say that these 11 genes or these 11 SNPs is just not a strong enough number to make our predictions. So let's look at our field-based visual selections that we made and plotted against their actual phenotype, their actual measurements. The R-square for 18 is 0.48 and for 19 is 0.55. Again, acceptable numbers, but you can see a better separation of top values than bottom values. Sure, there are outliers in the top 10% that we selected, but we can see a better separation of our fiber quality lines, the top producing versus not so better fiber quality lines. Same story is with 2019, much better separation of the fiber quality lines. For the bottom 10%, again, similar correlation numbers. There are values more towards the bottom 10% than there are towards higher upper half mean length values. Then again, field-based breeding has been working ever since cotton breeders have been doing it. So we should see a better separation of bottom 10% than the top 10%. For the visuals, sorry, the previous one was the interspecific population. This one is going to be the intraspecific. For 2018 and 19, for our field-based, really good R-square values in 2019. There was better separation in 19 than compared to 18. But then again, we can see that there is a better separation of the values. So for bottom 10% in 18, not a good prediction, not good selection that we were able to make that we thought would be based on our visual screening. For 2019, it was a better year. So visual screening, I guess, for 2019 had a better correlation in 19. So conclusions based on these 11 gene-based SNPs for upper half mean length. Our work suggests that maybe population with high polymorphism may be better suited because there's a lot more variation in an interspecific population. So perhaps it might be better suitable for a population with a wider range of upper half mean length values where we can select top and the bottom ones. Second, which I didn't show, was that for fiber bundle strength, there was a lack of robustness of these markers. Third one, which is obvious, that more genes should be, or gene-based SNPs are required for screening a breeding population for favorable alleles. And last one, which we expected, that field-based breeding was certainly better in separating the bottom and the top genotypes for upper half mean length and fiber bundle strength. And with that, I would like to acknowledge my committee members, Dr. Wayne Smith, Dr. Zhang, Dr. Hike, Dr. Rooney, and the Cotton Improvement Lab Don, Ali, and other graduate students and student workers, and for financial support, cottoning, and AgriLife research, and many others who have helped on this project. Thank you. Yes, Dr. Hall.
Let's just make a comment. Actually, we did a test the number of genes you need. When you do the gene-based breeding, we did from 10 up to 200, up to all 474. And you have to minimum need 20 SNP. Predict accuracy that's 0.4, 0.4, 0.5. If we use up to 200 as 0.85 predict accuracy. So that's certainly need an increased number of the genes in order to see the segment. That's actually we planned. That's the only things we are short in funding. We wanted to 100 or 200, but we don't have money now. And the second part that's you may use your data. You can improve. You only count additive effect. Every many of those genes, 10 of those genes, maybe there are few of those genes that's a dominant, over-dominant effect. So that's, you need to count. Heterozygote case, you can count 2 or 3 if heterozygote. If completely dominant, you count 2. If over-dominant, you count 3. That's the rule. You should check that because we did that test. If we count dominance additive both effect and predict accuracy much better than additive only. What you did only count additive. You didn't count dominant. We know which allele that's dominant, which one over dominant, which one that's additive. That's my suggestion. I wish you at least do the second part, second suggestion.
I used whatever you gave me. So. Sorry. Yes.
So just out of curiosity, you ran this one cycle?
One cycle, like 1 year of selections?
Well, or one cycle of selection, or
Yeah.
2 years of phenotype.
It's 2 years of phenotype. Yeah. So I repeated in 18 and 19.
But you only did one round of
Yes. Yeah. 1 round of selection. Yes. One in 16.
so I guess part of my issue is one round of selection is frequently very difficult to find significant differences. And so when you're comparing 2 techniques, that complicates it to have one round of selection. I mean, you've talked about how long traditional breeding has been working.
Yeah.
But seldom do we show a traditional breeding significance with one cycle.
I understand. Yeah.
We show it with 5 cycles or 10 cycles or whatever to get to where it's significant.
Yes. Sorry.
So in 2016, you had F2 population, and top 10 and bottom 10 were selected. These were individual plants.
Yes. Individual plants.
And how did you advance them? As a bulk or as individual to F2?
Individual to a progeny row.
So what was your test material? Because there will be large number of progeny. How did you advance them? From F2 to F3,
Yes.
there will be 10 progeny rows for the top and 10 progeny rows for bottom. Then for F4, as you further go, you take the bulk. How did you do it?
So in 16, we only had enough seed to make 1 progeny row.
Yeah,
For-
that would be 10 progeny rows.
No, we are.
Top 10%, let's say 10.
Yeah.
10% or whatever it is, you have so many progeny rows in F3. How did you harvest F3? Individual plant or bulk?
Oh, bulk. Yes.
So F4 and F3 were bulk.
Yes.
And for testing, final testing, how many plant samples did you
Oh,
test?
we did. Oh, there was 2 replications, so there were about 30 plants in each row, so you can say 60 plants total. There was 2 replications. We harvested the whole row for fiber quality. But we picked cotton from every single row. Yes, Doctor.
So I assume you have some data to show the selection differential and the sponsor selection.
Yes,
You have that data, right?
yeah, we have
Basically, you
done.
can actually compute realized heritability if you want.
Yes, yes.
That's one thing. The other thing is, did you validate the model?
Oh, we did the.
I didn't see the validation.
I only wanted to show whether I only wanted to show the prediction accuracy. We
How
have
did you validate it?
oh,
The verification of the model. Did you use bootstrapping or did you have actual data to validate?
well, what we did was from the 2 years of data, we ran an ANOVA on it, and then we determined the significance of genotypes, and then from their genotypes, we did a mean separation.
No, I'm talking about you have the predicted versus actual. So did you validate that model?
No, that
You can
validation.
use bootstrapping to do that.
Yes, sorry, I misunderstood. Yeah, the validation has not happened yet.
Can you go to slide number
4?
4? So with the two circular diagrams, can you explain the situation why it'd be better to have markers around the gene as opposed to the marker being the gene itself?
Well, here you have identified the gene, so you are sure of the effect of the
So
gene.
you always would want to have the marker be the gene itself.
I don't know. I mean,
How much more work does it take to have the marker be the gene or just get the marker close to the gene?
I don't know because I was not involved in this work. I mean, at least in the protocol. I think Dr. Hongren will be able to better answer that question. I think we're,
Yeah,
yeah.
Actually, that's I draw a circle. When you do the marker seed selection or genomic selection, first the based on genotype. And when you do a gene-based breeding here, you not only you do this all the sides of the circle that indicate all of the circles contribute to the traits. For example, the gene-gene interaction and gene action direction, positive or negative effect, and so on. That's a circle to contribute the phenotype of the trait. So that's gene-based breeding handle all those factors. But the gene-based breeding only marker seed selection or genomic selection only focus on the genotype of the lines or a locus.